Hello and welcome to Lab Down Under. In today's video, I'm going to be talking with Dr. Sebastian Duchene of the University of Melbourne about an opinion article he wrote on phylodynamic methods for bacteria. So phylodynamic methods have been predominantly used in fast evolving things like viruses and bacteria to see how the gene changes, to see how mutations, the speed at which mutations come, the speed at which these things are able to spread. And traditionally, these methods have been used more so in viruses, which have a simpler genetic structure than bacteria. And Dr. Duchene's opinion article was basically highlighting that gap and saying, hey, we should be focusing on bacteria too, because these things may create some kind of emerging threats in the future. For instance, you might have a more virulent strain of bacteria emerging. You might have different types of bacteria emerging, and these are things that we generally should be looking at in the future. So it was all very, very interesting, and I hope you enjoy the interview. Today I'm speaking with Dr. Sebastian Duchene of the University of Melbourne about his latest work on phylodynamic methods and bacteria. So hello, welcome, Dr. Duchene.
Yeah, thanks for inviting me.
Yeah, no worries. I guess, so we're here to speak about your latest opinion article looking at this topic. But I guess before we do dive into the article itself, maybe you could explain a little bit about the topic itself, phylodynamic methods, and what exactly that kind of means.
We have problems in all the phylogenetic and phylodynamic. Like whenever we talk to the public about this, the words are a bit hard even to pronounce. But in general, phylodynamic methods stem from a range of methods called phylogenetic methods. And phylogenetics was designed to understand evolutionary relationships between species. Mm-hmm. For instance, if you look through Darwin's old notes, you would find a phylogenetic tree. So this is just a family tree that connected different species. And we've been using these methods for a while now to understand the evolution of different organisms. But in pathogens, because they evolve so quickly, there's actually a huge amount of information in their genomes that we can exploit in phylogenetics. So much so that we can even infer things like how quickly they're spreading in a host population, how fast they are accumulating mutations. Mm-hmm. And this melding of epidemiology and molecular information is known as phylodynamics. These methods are very well developed in viruses. We've been using them for over a decade now, and they've played a big role in how we understand the current pandemic. But in this opinion piece, what we thought is, there's a lot to do in the bacterial space as well, where we have a lot of data and a lot of metadata associated with them.
So can you just explain then, in the article itself, you looked at phylodynamic methods in bacteria. So what did you look at, and kind of why did you decide to look at this? I mean, you mentioned that there was a gap, that this was done predominantly with viruses before, and that it hadn't been done with bacteria now, right?
Mm-hmm. So we identified, or basically phylodynamics in viruses has had three main fields or themes that people have investigated. One of them is molecular evolution, and that is how quickly you accumulate mutations and whether those mutations confer resistance to drugs or an escape from the host immune response, known as antigenic drift, mm-hmm. or escape from vaccines, that sort of thing. So that molecular aspect. The other aspect that has been developed quite well in viruses is looking at epidemiology. So we can use virus genomes to understand or to estimate a parameter that is now very famous, the reproductive number. Mm-hmm. We can estimate that using virus genomes, and also how quickly they move between perhaps different locations. So if you've got a virus here in Australia and in America and Europe, we can estimate the rate over time at which they have moved using the genomes. Mm-hmm. And that's kind of known as phyloepidemiology or phylogeography. So those are kind of just the broad topics in phylodynamics. And the reason why they were developed in viruses is because viruses have much smaller genomes than bacteria. So it's very easy to sequence the complete genome. And also because they evolve much faster, it means that you get all these mutations in the genome that we can exploit using these statistical methods within phylodynamics. What we argue here is, well, many bacteria cause a lot of important diseases. Like tuberculosis still kills lots of people every year. We have emergent outbreaks of Shigella, E. coli, and many others. The genomes, even though they're very difficult to sequence, technology has improved dramatically. So in some cases, we have thousands of genomes for bacterial outbreaks in different parts of the world, and very good associated metadata. So our argument here is, well, the data are there. They are more complex, but we can analyze them using these phylodynamic methods. And that holds promise for us to understand the emergence of perhaps lineages that might be more transmissible, like we're seeing with coronavirus and the variants. We could do that in bacteria, and also understand what drives the emergence of antibiotic resistance or drug resistance in general, which is a big problem in bacteria these days. So those are questions that we think could be addressed using phylodynamics. And we haven't quite got there just because there's a few methodological hurdles, notably with computational speed and also developing the right statistical methods to analyze these data.
Yeah, that's what I wanted to ask then. Was it just because of the complexity that we haven't kind of looked into the bacterial genomes as opposed to the viral genomes, which you mentioned are a little bit, are a lot simpler to understand? So it was just that complexity, right, that was kind of holding up everything?
Yes, mostly. There's a few caveats to bacteria. First, because the genomes are larger, you need more computational power to analyze them in general. The second is that viruses reassort, that is, they exchange genetic material, but less so than bacteria. In some bacteria, the primary mode of evolution is not necessarily mutations, but recombination. So that's where different bits of the genome break apart and then get stitched together in another bacterium. That means that if you assume that evolution is tree-like, then your data don't meet that assumption at all. Recombination means that you can't use standard phylogenetic methods. And I think that's been the major problem with analyzing bacterial data using these frameworks, and the reason for why we have such good methods for viruses, but less so for bacteria. Of course, there's very good people working on these problems, but yeah, I think that recombination would be the main problem.
So you've said the paper basically said about the why, and you've mentioned this as well, was to combat different emerging threats. So antibiotic resistance, you found more. Do I use the word virulent strains for bacteria, or is there another or more pathogenic?
No, you can. Yeah, yes. Virulent is just how pathogenic they are.
Yes. Okay. So yeah, was it just those two kind of threats, or was there anything else that we could, that this kind of research could be used to detect?
Well, it's very difficult. Yeah, drug resistance is probably the most pressing threat in bacteria, but it's also possible that new bacteria can emerge. For instance, here we talk about bacteria that have environmental reservoirs, like Klebsiella is one of them, where we don't necessarily have human-to-human transmission, but it gets transmitted from the environment. So depending on what the environment is, if you're more exposed to perhaps soil where the bacterium lives, then that's more likely to cause an outbreak. But these are things that we can identify by statistical methods, finding out whether perhaps contact with a certain environment predisposes you more to acquiring, to getting a bacterial-associated infection. So I think changes in environment is also things that we need to investigate. There's a few reasons for why some diseases might be more common or might spread more. In the past 60 years, where we have interacted differently with the environment.
So, what were the kind of limitations to how we could use these phylodynamic methods with bacteria then?
I think, well, the first thing that we need to do is to have a good understanding of what we're sampling out there. With viruses, it's quite easy, particularly, say, in Australia, the way the coronavirus pandemic has been handled makes that aspect of the analysis quite straightforward. We know that we're obtaining genomes for about 90% of all positive cases. With bacteria, it's less clear. If some of these infections cause no symptoms, or have very long latency period, then we're not sure what the strategy is. So that's one thing that we need to understand better. And to that end, I think it's a matter of coordinating research and data collection efforts a bit better. Yeah, so I think that's one limitation. The other one is really understanding how to use computational methods for this. For instance, you mentioned recombination earlier. We most methods that deal with recombination are quite slow. So if you want to infer the evolution of the whole genome, including those bits that have been recombining, you're going to run into a lot of problems if you have a reasonably sized data set, say 1,000 sequences, 1,000 genomes. Instead, what we tend to do is we'll just remove whatever recombining regions are in the genome. So we get rid of a lot of data and then use a subset of it. But of course, that means you lose a lot of information. So that's another thing that we need to work on, how to include this information effectively, how to exploit it computationally, and just to understand better what we're sampling, which can partly be addressed via epidemiology. There's also one implication, and that is the transmission dynamics are a bit weird. For instance, if you have an environmental reservoir, an animal reservoir, and human infections, then you need to design an epidemiological model where you have transmission between these different, we refer to them as deans, these different kind of compartments. And it might be that between humans, you have no transmission whatsoever, but between the environment and humans, it might be quite common, or environment and animals and back and forth. So parameterizing and designing those models properly is another hurdle that we have to go through.
Finally, then, you've written this piece, and you've mentioned all the problems that we need to tackle. So what do you want? What do you want people? What do you want scientists to do? What do you want laboratories to do? What are the kind of flow-on effects now that you want from now you've written this article and it's being spread around in the community?
Yeah, so I would like to push people doing virus phylodynamics to collaborate more with people doing bacterial genomics. People doing bacterial genomics understand their bacteria extremely well. If we talk more to each other, then I think we'll be able to develop better methods. But sometimes we're very much fixed on the one bug that we work on. This particular collaboration, I think, is quite neat. I come from a very virus background. I only worked in bacteria until recently, and not in a lot of depth. The first or second authors, especially Danielle, she's a big expert on bacterial genomics. So talking to her, we've collaborated on a few projects where she'll tell me, well, this is the biology of this organism. This is what the genome looks like, and this is what you can take into account. Then I'll model it somehow. And it might happen, it often does actually, that my model makes some assumptions that are not quite valid for the bug that we're studying. So I think promoting these conversations, making the research a bit more interdisciplinary would be great. I hope that's what this article will promote.
And is that what kind of prompted the article itself? The prior collaboration with Danielle and the kind of issues that arose from that? Yeah.
When I arrived at Melbourne Uni from Sydney, I was only doing virus stuff, and then when I got here, I thought I could just translate that easily into bacteria, and that wasn't the case. I just found it extremely difficult, so I actually didn't want to work on bacteria. But then talking to people like Danielle, Ben, and others at the Doherty Institute, I realized, okay, we can do this, but we just need to take a step back and rethink our models a bit better. But we did see the potential of these collaborations. It's hard work.
Yeah, which is another hurdle, which you really step over, I guess.
Yeah, absolutely.
Yeah, but no, I mean it's definitely very, very important to do. All right. Well, I mean, thank you very much for this, Sebastian. It's been good, and I wish you, the best of luck. I hope your article kind of prompts the kind of collaboration and new findings that you want.
Thank you. Yeah, my pleasure.
No worries. All right, Sebastian, we'll be in touch.
Thank you. All right, talk soon. Bye.
Well, I hope you enjoyed that interview. If you would like to find out more about Dr. Duchene's research, I'll put a link down below to the Lab Down Under blog article, which I'll write about this, and I'll also add some links there to Dr. Duchene's social media pages, university pages, and stuff like that, so you can find out more there. If you would like to, support this article, support the blog, please don't forget to like the video, subscribe, press the little bell icon, leave a comment below if you want, and of course Lab Down Under is on a ton of other social media platforms as well, which I will also link below, such as Twitter, Reddit, Facebook, and LinkedIn. We also have a Patreon page as well if you would like to financially support the blog, and of course that will always be appreciated too. So until next time, until the next video, keep on being curious, and I'll catch you later.